until very recently, or other genetic conditions such as hereditary hemochromatosis. So genetic factor influencing the development of liver fat accumulation are a major determinant of the risk of this condition. And the main one you are probably aware is the variant in PNPF3, which is the main risk factor for fatty liver steatohepatitis, so that it could be incorporated in non-invasive scores, for example, of fatty liver and NASH, allowing to discriminate with moderate accuracy the development of this condition to estimate, for example, the prevalence of NASH in large population studies. So this is a main risk factor for this condition. So given this important role of genetics, in particular some specific genetic factors in pathogenesis of this condition, it was proposed that this can be used to develop a personalized approach also to the clinical management of fatty liver disease, moving from a traditional healthcare model where the same treatment is applied to all patients affected by this condition, fatty liver disease, to a targeted approach where in the future we will hopefully be able to implement a specific treatment in patients with some molecular features, so that we can administer a drug that is effective and not toxic to the patient, avoiding the risk of administering it to patients where it is toxic or where it is not effective. So the main risk factors for this condition turn out to be related to mostly the management of lipids within the key cells in the liver, that is the hepatocytes, starting from the uptake of glucose from the blood, the regulation of lipogenesis, secretion of very low density lipoproteins by the liver and lipotoxic pathway, but in particular, the culprits for increased risk of this condition were identified at the level of the remodeling of lipids at the surface of lipid droplets in the hepatocytes. And you know, as I anticipated you before, the main one is this common variants in this gene called PNPLA3, encoding for the ileosin for methionine substitution at position 148, when the wild-type protein is expressed at the surface of lipid droplets in the hepatocyte that is induced during obesity and insulin resistance in response to insulin. It is involved in the remodeling probably of fatty acids and phospholipids in the lipid droplets. It is thereafter ubiquitinated and degraded by the proteasome. On the contrary, the methionine variant of PNPF3 is not enzymatically active. It accumulates at the surface of lipid droplets where it cannot be ubiquitinated, and at this level, it acquires the ability to sequester a main cofactor for ATGL or PNPLA2, the main lipid hydrolase in the lipid droplets of hepatocytes, therefore causing a reduction of lipolysis and entrapment of lipids within this compartment in the hepatocyte, development of progressive fat accumulation and lipotoxicity, triggering then steatohepatitis and fibrosis. And this is very important because, as I told you before, this has a huge clinical impact in not so, sorry. Okay. I had a problem with the slide. So it has a clinically significant impact in the population because if you look at the prevalence of the at-risk genotype in the population, in particular homozygosity for the methionine variant, you can see here from the scheme, then in the European population, 1 in 20 individuals is homozygous for these genetic factors, but the prevalence of the genotype increased progressively with the severity of liver disease across the spectrum of fatty liver disease, and about almost 1 in 2 individuals with hepatocellular carcinoma due to NAFLD, but for alcoholic liver disease, the situation is very similar, is homozygous for this variant. So this is a 9-fold absolute increase in the risk of finding this genotype, which is a huge genetic effect on this condition. Is very similar to the increase in the risk of liver cancer, which is conferred by the classical mutation of hereditary hemochromatosis, which is a well-known genetic cause of liver disease. So this first phase of the genetic discovery allowed to determine that when there are triggers for fat accumulation in the liver, obesity, insulin resistance, impure or metabolic dysfunction associated NAFLD, but also excess alpha intake, dietary fructose intake, chronic hepatitis C virus infection, the methylene variant of PNPF3 will favor the accumulation of fat in the liver, favor lipotoxicity and development of steatohepatitis, and then lead to cirrhosis and also to hepatocellular carcinoma. So what we found then is that the impact of the mutation on liver carcinogenesis was also partly independent of the effect of fibrosis, because it was also evident in patients with cirrhosis independently of the severity of liver damage. And the fact is so important that actually this is also associated with an increased rate of mortality in our patient with liver disease. You can see here data from an Italian core study of patients with NAFLD in Sicily from the group of salvopetta. You can see here that the number of at-risk allele in PNPF3 was associated with an increased risk of mortality related to liver events in patients with NAFLD, and carriage of this genetic risk variant was associated with mortality for liver disease in the NANAS free study in the US population. You can see here that the liver disease mortality was increased by more than 8-fold in individuals homozygous for the methylene risk variant in the US population, which is a huge increase in the risk of liver disease. And this was also associated with an increased risk of overall mortality in the general population, including this study, which was on average overweight, therefore a high risk of developing fatty liver disease. So this is the main determinant of the risk of progressive liver disease, even considering the acquired risk factor for the infection, for this condition, sorry. So during the last years, we also identified other genetic risk variants for fatty liver disease. And in this case, we started from evidence a few years ago that this variant in these Ambos7 genes is an independent risk factor for development of alcoholic cirrhosis, which emerged from a GWAS study. And we show for the first time that this is also associated with an increased risk of hepatic fat accumulation in the general population, again, and in the Dallas study conducted in the southern US, where hepatic fat was quantified by non-invasive gold standard by nuclear magnetic spectroscopy. And this association with fat accumulation also translated with an increased risk of developing steatohepatitis, this is the progressive form of fatty liver disease, and also liver fibrosis. So what we have very recently confirmed in a collaborative large international effort which encompasses individual patient data meta-analysis of very huge number of individuals, more than, for example, 21,000 in this case for the hepatic fat accumulation, is that indeed this represents another risk factor for hepatic fat accumulation in the liver, increase the risk of steatosis, and this translates in increased risk of developing NAFLD. Again, you can see very large numbers, more than 200,000 individuals, and increased risk in particular severe fat accumulation in those who underwent liver biopsy, leading to an increase in the risk of cirrhosis and in the risk of hepatocellular carcinoma. So this is another risk factor for fatty liver, which leads to progressive liver disease. This is important because it's another gene which is involved in the remodeling of lipids, and in particular phospholipids at the level in the plasmic reticulum, including in hepatocytes. In particular, Ambot7 is involved in conjugating a molecule of arachidonic acids to lysophosphatidylinositol in the liver to produce phosphatidylinositol bound to arachidonic acid within the so-called lens cycle, where the saturated phospholipids are desaturated within the liver. And we first showed that this variant is associated with reduced expression of Ambot7 in the liver, leading to an increase leading actually to a decrease in the circulating level of phosphatidylinositol bound to arachidonic acid in the circulation, which was later confirmed in the liver, and it was later also confirmed in an experimental model of fatty liver disease as well in other cohorts. You can see here in this recent paper, which has just been published in GAT, to which we have collaborated together with Stefano Romeo and Japanese group, which confirmed that in individuals carrying the genetic risk variants for NAFLD, PNPF3, there is a decrease in circulating phosphatidylinositol bound to arachidonic acid. And this was recapitulated in mice models where Ambos7 was specifically knocked down in the liver. So therefore, the risk variance for fatty liver disease is associated with a reduction in the function of the enzyme, leading to a decreased production of phosphatidylinositol bound to arachidonic acid, and on the other hand, an increase in the substrate of the reaction, namely lysophosphatidylinositol species, which may have an important role in the pathogenesis of liver disease. What we have also recently I highlighted is that the genetic background is not the only determinant of the expression of Ambos7 in the liver, because this downregulation of Ambos7 is not only due to genetic risk factors for liver disease in the Ambos7 gene itself, but it's also associated with the severity of liver damage, in particular inflammation, as shown here in this cohort of patients with severe obesity, where we analyze gene expression, but in particular in this and another cohort with the presence of diabetes and the severity of insulin resistance. This data have also been confirmed by independent groups, such as here. You can see here that Ambos7 is dramatically downregulated in obese individuals as compared to healthy subjects, and in experimental models as well. There is a downregulation of Ambos7 expression during fatty liver disease, which can be due to obesity, such as in the ep.1 mice, or to high-fat diet. And the degree of downregulation of this Ambos7 correlates with the severity of insulin resistance as estimated by body fat mass, or the OMA, for example, insulin resistance index. So we recapitulated exactly these results in our lab, showing that, for example, Ambos7 is downregulated not only in dietary models of steatohepatitis due to the methylcholine deficient diet, but also in particular in ep.1 mice, where there was not only a downregulation of the gene expression, but also the expression of Ambos7 at protein levels. And we tried to study what were the mechanisms leading to this downregulation of Ambos7. So we first observed that Ambos7 is downregulated in response to refeeding following fasting. So it's regulated depending on the nutritional state in mice in response to the increase or in parallel to the increase in insulin levels following the feeding, which is, of course, paralleled by an increase in activation of the insulin signaling pathway in the liver with increased level of phospho-AKT to AKT ratio, phosphorylation of FOXO1, and their consequent nuclear exclusion and downregulation. This process followed by progressive downregulation of FOXO1, Ambos7 expression. But this process was even more marked, recapitulated by the intraperitoneal injection of insulin, suggesting that this directly dependent on the insulin signaling activation, irrespective of nutrient state or the availability of glucose. Indeed, during this experiment with insulin tolerant test, you can see here that there is even more marked activation of the insulin signaling pathway. Phospho-AKT increases in very short time in this model, as highlighted here in this Western blotting. And this is associated with a marked decrease in expression of Ambos7. You can see here this Western blot. So to make a long story short, we have conferred this data in a cell autonomous manner in primary hepatocytes in mice. Even here, exposure to high dose of insulin led to a downregulation of Ambos7 in a PI3 kinase dependent manner. The downregulation was completely inhibited by the pre-incubation with PI3 kinase inhibitor, whereas the upstream kinase AKT phosphorylation was not, suggesting that this was a mediator of the process of downregulation, probably exerted at gene expression level by FOXO transcription factor. So in this way, we hypothesized that the hyperinsulinemia typical of this metabolism can lead to downregulation of Ambos7, especially in carriers of the mutation, that this may favor hepatic fat accumulation. And to further prove this hypothesis, we've induced an acute downregulation of Ambos7 expression in the hepatocyte of mice by injecting them with antisense oligos, leading to about 50% decrease in hepatic protein expression, as shown here. And this goes in the following day, and about 50% increase in hepatic fat accumulation, leading to the development of microvesicular steatosis in this model. Actually, this was confirmed by independent groups, for example, in this paper by Japanese group, showing that the liver-specific Ambos7 knockout causes fatty liver disease by, again, impairing lysophosphatidyl inositol remodeling. Again, here you can see that the liver-specific knockout of Ambos7 causes a depletion in phosphatidyl inositol bound to arachidonic acid, whereas on the other end, there is a corresponding increase of species bound to fatty acid, and this leads to an increased accumulation of fat into the liver, leading to steatosis, which is partly also dependent on the upregulation of genes involved in lipogenesis, such as SCD1 and fatty acid synthase in this model, whereas the secretion of lipids in the liver is not affected. So the main mechanism hypothesized for this effect of the downregulation of Ambos7 based on these studies and in vitro cellular studies models we've developed by knocking down Ambos7 by CRISPR-Cas9 in ep.2 hepatocyte is that the accumulation of fat into the liver depends by the shunting of phosphatidyl inositol, which cannot be remodeled to the synthesis of deacylglycerol, which is then converted into triglycerides, and by the upregulation lipogenesis, which may be related to upregulation of the mTOR pathway. And another possibility is that was highlighted by the Japanese group is that the reduced amount of phosphatidylenosidol bound to arachidonic acid in the hepatocytes will not be able to inhibit CDS, leading to an upregulation of the synthesis of CDP, the acylglycerol, and the synthesis of triglycerides by this alternative pathway. So whatever the relative role of the specific pathways, we and others show that the regulation of Ambos7 during insulin resistance in the liver is associated with an increased accumulation of fat into the liver, and this is probably the main mechanism by which it causes progressive liver disease. Probably is not the only one, because this process is also paralleled by, as said before, an accumulation of the substrate of the reaction, lysophosphatidylinositol. And there is initial data in clinical studies that the levels of lysophosphatidylinositol bound to saturated fatty acid are increasing in patients with advanced liver disease, whereas there is a decrease in phosphatidylenosidol bound to arachidonic acid, consistent with the effect in the Ambos7 enzymatic activity. This was parallel in mice where the hepatic Ambos7 expression was silenced by antisense oligonucleotide, similarly to the models developed in our lab I showed you before. And what was important that in the presence of hepatic Ambos7 downregulation, the exogenous administration of lysophosphatidylinositol was able to induce genes involved in triggering hepatic inflammation, such as TNF-alpha, IL-1 beta, and to activate fibrogenesis in the liver, as shown here by the expression of actin, collagen, and so on. And these mice developed, in particular, similar to those with liver-specific knockout of Ambos7 progressive liver fibrosis. So actually, this effect of lysophosphatidylinositol on the particular deactivation of fibrogenesis may be mediated by a direct interaction which with a specific receptor, which is GPR55, is an endocannabinoid receptor which show a specificity for lysophosphatidylinositol, and it is involved in the activation, in particular hepatic stellate cells. This is a recent paper showing, published just the end of last year, showing that lysophosphatidylinositol is able to induce liver fibrosis in mice, and activation of LX2, which are in vitro models of hepatic stellate cells. And this process can be inhibited by the silencing of GPR55 in hepatic stellate cells, suggesting that this is a receptor-dependent process which is mediated by this specific pathway. So what are the implications of the discoveries on what we know on the pathogenesis of fatty liver disease, and how can we exploit these findings to develop new treatments and new clinical management strategies for this condition? An important, I think, observation that we and others have made during this phase of discovery is that the impact of these genetic risk variants on the progression of liver disease, and in particular on fibrosis, which you know is the main prognostic determinant of liver-related events in patients with non-alcoholic fatty liver disease and fatty liver disease in general probably is directly proportional and very closely related to their impact on hepatic fat accumulation. So in this slide, you can see that it's plotted the impact, the normalized impact of the main genetic risk variant known a couple of years ago to influence hepatic fat accumulation on the x-axis on steatosis, histological steatosis, while on the y-axis there is the impact of fibrosis. You can see here that there is a nice linear correlation, which is very consistent, and suggesting that the main mechanism by which this genetic risk variant leads to progressive liver disease is by determining a quantitative and qualitative alteration of hepatic fat in the hepatic, so that the hepatic fat accumulation is not an innocent bystander in the pathogenesis of fatty liver disease, but plays a causal role in determining progressive liver disease. So this can be formally tested in the Mendelian randomization approach, and it was demonstrated by our group. But what's also important that a similar relationship can also be drawn for the risk of progression to hepatocellular carcinoma. Again, here you can see on the x-axis there is the impact of the main genetic risk variant for fatty liver disease on hepatic fat accumulation, while on the y-axis the impact of on the risk of hepatocellular carcinoma in a large collaborative European course. So you can see here that again that there is a direct and linear relationship between the impact on the risk of fatty liver disease and then the progression to hepatocellular carcinoma, which is also partially independent on the effect of liver fibrosis. And by a range of analytical approach, which is not very easy to explain in a short time, we show that this association is consistent, if not yet a final proof, with a causal relationship between hepatic fat accumulation and hepatic carcinogenesis, which is also evident in those without liver cirrhosis and independent of the effect of cirrhosis. So if anyone has some question, this I can answer during the discussion. So the main mechanism leading to this progressive liver disease associated with hepatic accumulation of fat within the intracellular lipid droplets may be due to the reduced ability to incorporate free fatty acid and other lipotoxic compounds and these intracellular organelles where they are less toxic by promoting lipoperoxidation, oxidative stress, and again, lipotoxicity. Maybe due to the initial consensual accumulation of lipotoxic compounds, including some species of phospholipids, probably free cholesterol, for example, ceramides, or the direct activation of stress pathway may be related, for example, to the accumulation of ceramides within the liver. So that the hepatic fat accumulation in the liver, fatty liver disease may be compare or to the atherosclerotic process to summarize the process leading to liver damage, whereby the initial phase would be related to accumulation of fat at the intracellular levels, in this case, the hepatocyte triggering lipotoxicity, oxidative stress, inflammatory pathway. And with time, this will lead to progressive fibrosis in targeted organs and the development of organ-specific complication. But we may also be more close to an application of these genetic discoveries to the an improvement of the risk stratification of patients. So as I show you at the beginning, the identification of PNPF3 variant was helpful in developing non-invasive scores able to estimate the prevalence of fatty liver disease at population level, but this was not yet sufficient to develop accurate score able to discriminate the risk of progressive liver disease at the individual levels. But with the identification of new genetic risk variant, we are improving by the day the ability to predict fatty liver disease now. So for example, here by adding two PNPF3 variants in TN6SF2, which is involved in the secretion of very low density lipoprotein, the retention of lipids in the hepatocyte, Ambos7, we already observed that the number of risk alleles at this loci was associated with a progressive increase in the risk of developing fatty liver and hepatic fat content in this population study, again, the Dallas study. And in European collaborative cohort, this same very simple instrument, the number of genetic risk variant in these genes was associated with a progressive increase in the risk of developing hepatocellular carcinoma, which was independent of classical risk factor, even independent of severity of fibrosis. And more recently, by combining the 5 main genetic risk factors for this condition, besides the three I mentioned before, also variants in glucokinase regulator, modulating the lipogenesis in the liver, and HSD7TB13, which is modulator at the risk of lipotoxicity, inflammation in the liver. We developed polygenic risk scores of hepatic fat accumulation and fatty liver. And in particular, we show that both in risk individuals in large European cohort and in the population-based study, the UK Biobank cohort, carriage of having a high genetic risk of fatty liver disease, a high polygenic risk score, which is observed in about 1 in 12 individuals in the general population, carries a risk of developing fatty liver in the presence of risk factor, which is about 3-fold higher than those who are negative, and also to progress to cirrhosis, increase the risk by almost 3-fold, and also in patients with cirrhosis to progress to hepatocellular carcinoma with an increasing risk by about 2-fold, including in those who go on to develop hepatocellular carcinoma in the absence of cirrhosis and advanced liver fibrosis. So we submitted that the use of this polygenic risk scores may be useful in the future to identify those individuals with this metabolism for whom intensified lifestyle management, additional workup, enrollment in clinical trials can be cost effective, and who probably should be prioritized in a resource-limited setting for ACC surveillance. So if I have probably 5 minutes again, I will show you how the situation is currently changing at a very rapid pace, because during the last year, the availability of large population study with genomic data, which were released for analysis to independent investigators, allow us to identify new risk variants for fatty liver disease, as other groups as well. Actually, this is a recent study, for example, that was just published in gastroenterology, which I collaborated again with Stefano Romeo's group, and the concept here was to identify the risk variant for ALT levels in the UK Biobank cohort, which is a very large cohort of more than 500,000 individuals enrolled in the UK. And we look specifically for variants determining alteration in protein sequence in this population cohort. So here the remarkable findings was that we confirm as the main determinants of ALT levels in the population, and it was already known variants in PNPF3, for example, and TS6SF2, which are the main genetic risk factor for fatty liver disease. We also observed the impact of HSD17B13. I showed you before, it's an important determinant of fatty liver disease and has positive control, for example, the alpha-1 antitrypsin main mutation as well. But we identified almost 91 new loci associated with increased ALT level. And what's remarkable was that the majority of these risk variants were located at genes involved in the regulation of lipid metabolism, consistent with the fact that alteration in hepatic fat accumulation and lipid metabolism are one of the main drivers of liver damage at the level of population levels. So, for example, here I show you this pathway analysis showing that among the most represented pathways among these loci associated with ALT level were lipid homeostasis and the synthesis of acylglycerol and their catabolism, in particular within the liver. There was, again here, biosecretion and PIA3 AKT signaling pathway involved in insulin resistance. And among the diseases, metabolic syndrome, fatty liver, steatohepatitis, liver disease, cholestasis is another hit, so probably modulation of the bile acid metabolism in the liver is another trigger, which also here has emerged the main driver of progressive liver damage in the population and lipoprotein metabolism in general. So in particular, we found two variants that besides ALT levels were also independently associated with significant association at genome-wide levels with hepatic fat accumulation in a subset of about 50,000 individuals with hepatic fat content evaluation in the UK Biobank, and we further demonstrated that in a larger European liver biopsy cohort, that these two genetic risk variants were also associated with hepatic fat accumulation at the histological levels. These are two variants in GPAM, which is involved in the synthesis of glyceraldehyde phosphate and fatty acids in the liver, with the risk variant associated with an increased risk of fatty liver disease. And the other one, interestingly, is the ApoA4 variant, which is the main risk factor for genetic risk variant for Alzheimer's disease, which consistently with other data that are coming out, resulted protective against not only LT levels, but also hepatic fat accumulation in the UK Biobank, the histological steatosis, remarkably also against the development of cirrhosis and liver cancer in the same cohort. So an interesting result is that the main risk variant for Alzheimer's disease and major predictor of dyslipidemia in the population actually protects against liver disease by reducing hepatic fat count. And by combining all these genetic risk variants together, the main ones I showed you before, plus these two novel, again, we were able to stratify the risk of hepatic fat accumulation quite nicely in this population, being able to predict the hepatic fat content with a good level of accuracy. Here shown the p-value, there is not a discriminant value, but the main concept here is that by discovering new genetic variants with for this condition, we are improving the ability to discriminate the risk of progressive fatty liver disease. In the future, combining rare variants with a large effect on lipid metabolism may also be useful. I've shown you these results just to report you that by performing whole exome sequencing with resequencing of candidate genes involved in lipid metabolism, liver disease, for example, we show that the carriage of ApoB mutation, again, reducing circulating lipid levels and determining compartmentalization of fat into the liver, resulting in higher risk of developing cirrhosis and liver cancer in an Italian cohort of patients with advanced liver disease. And combination of multiple genetic risk factors, including the carriage of the ApoB variant, was able to improve the ability to discriminate the risk of developing liver cancer, achieving discrimination which was not yet optimal, of course, but better than the PNPF3 variant alone, or as shown in blue here, or a combination of the main common genetic risk variants of the condition. So in the future, the combination of multiple genetic risk variants in polygenic risk scores may be able to improve the ability to discriminate the complication of fatty liver disease over that possible based on classical risk factors alone, such as age, sex, BMI, type 2 diabetes, and fibrosis biomarkers, in particular in non-invasive setting, which is the future for clinical referral pathways in this condition. So a final note on a possible Impactor therapy. Carriage of the PNPF3 variant is associated with an increased risk of liver complication. I show you this very clearly, but is also associated with a different pathogenesis with reduced remodeling of lipid droplets as compared to the novel hypogenesis. So could be speculated that therefore the silencing of PNPF3 proteins in carriers of this variant, which is a large fraction of those who are at risk of progressing to severe liver disease, may be able to reverse the molecular defect leading to retention of fat into the hepatocytes and ameliorate liver damage, reducing the risk of developing complications in these individuals. So again, this has been proven in experimental models in mice fed steatogenic diets, the high sucrose diet here, or a westernized diet here above, showing that in particular in mice engineered to express the mutant variants of PNPF3, the silencing of PNPF3 in the liver by delivering antisense oligonucleotides conjugated by with glucosyl residues, was able to inhibit not only steatosis accumulation, shown here with an effect which was more marked in mice carrying the mutation as compared to wild-type mice, but in particular in mice on Western diet, this approach was able to reduce liver fibrosis. Here, fibrosis is shown here by collagen immunohistochemistry. You can see that this here, in particular in mutant mice, there is a nice reduction, whereas fat accumulation was shown here by oil redox staining in red. So the idea is that this identification of fatty liver disease variants such as PNPF3 may lead to, by means of functional studies and experimental models, to identify approaches able to interfere with the disease pathway, which for PNPF3 may be related to the remodeling of lipid droplets, alteration in retinal metabolism, and activation of inflammatory pathway, and to test this approach first in experimental models of the disease, and then finally in clinical studies in patients, leading to the development of pharmacological approaches which may be very effective in patients with a distinct fatty liver phenotype with a high likelihood to respond to these approaches. At the same time, the development of a risk stratification tool may help to select those patients who are candidate for this precision medicine clinical approaches, but also for the current but also those who can be prioritized for what are the main approaches that are currently held: surveillance for liver cancer, inclusion in clinical trials, and intensification of lifestyle management. So this was my last slide. This is a recap with the main conclusion, and thank you for your attention. Sorry if I was a little bit late, but I would be very happy to answer a question if there is time.
Well, thank you very much.